Thank you, David. And I must say it's nice to rejoin this Sophian family. Unfortunately, I had no problem getting a visa from London. So, well, I'm going to talk about the emperor of all melodies, cancer, as somebody called it. Pointer? I should have done this before. Okay, so we're going to talk about the pathophysiology of specifically voltage-gated sodium channels in cancer. And specifically, I want to present to you for the first time what we put together as the in vivo evidence. Now, here is the plan of my talk. I'll do an introduction, some principles, laying the ground. We have a specific hypothesis that we're developing, and then the bulk of the lecture covering the in vivo evidence. And I'm going to do this hierarchically from mRNA to clinical studies. So we'll go through evidence at mRNA level, protein level, tissue electrolytes, animal models, and so on, and then we'll conclude. Now, it's well established that all ion channels are in all cancers. So it's just like brain. This just summarizes all the different channels in different cancers. So take it away that all ion channels are all in cancers. And then the question becomes, what to focus on? What channel to adopt and what aspect of this complex pathophysiology to investigate? Well, we have put the money on metastasis. So metastasis is the process where cancer spreads, and this is the main cause of death in cancer patients. As long as a tumor stays where it arises, it is not often life-threatening, and it can be surgically removed and so on. But once it spreads, once it metastasizes, it becomes much more difficult to manage clinically. And so even though this is quite a complex process, as somebody equaled it to the complexity of development, embryonic development, we can actually, in a reductionist way, reduce it down to a series of what I call basic cellular behaviors like motility and secretion and adhesion and obviously cell cycle and so on. And we know very well that ion channels are involved in all such basic cellular behaviors. So in a way, it was logical in hindsight to look for ion channels in metastasis. So this summarizes the metastatic cascade. So the whole thing may start with the growth of a primary tumor, bless you, and then at some point, the tumor cells within this primary tumor, the cancer cells, through some proteolytic activity, will break out through the basement membrane and then get into the circulation. And then once they spread around, metastasis occurs. Now, there are two things I want to highlight here that are relevant to my presentation, one more than the other. And one is the fact that growing tumors become hypoxic inside. So they start lacking in oxygen because blood is not flowing effectively. The other major problem, which I'm not going to deal with in any detail today, is the occurrence of these so-called micrometastases. These are dormant metastases which do not develop angiogenesis. They just sit there like in hibernation, and they're very difficult to detect clinically. And these are the guys that often lead to cancer recurrence and a major headache in the clinic. But hypoxia is very important for us pathophysiologically. Okay, so when we ask the question, what are the ion channels involved in metastasis? We know all about these basic cellular behaviors. We and others have mapped out a kind of profile of voltage-gated currents in tumor cells of various metastatic potential, or various degree of aggressiveness. So these are all human breast epithelial cells, going from normal through benign to all the way to aggressive and metastatic. And when we look at profiles like this, we did this originally, in fact, for prostate. We broadened it out to breast. It's also been seen in some lung cancers and also in ovarian cancer and so on, what we see are two things. First of all, these outward currents clearly get smaller and smaller as the cells become metastatic or acquire metastatic potential. And eventually, the cells start expressing these sharp inward currents, which, of course, a trained neuroscientist, and I did 25 years of neuroscience research, will recognize it as the activity of a voltage-gated sodium channel. And that's been confirmed pharmacologically by toxins and a lot of other techniques as well. So from this, we put together what I called the CellX hypothesis, the cellular excitability, which says in order to metastasize cells, cancer cells upregulate their voltage-gated sodium channels, they downregulate their outward currents, mainly potassium. Of course, these would normally be inhibitory, so they lose their inhibition, and together the membranes become excitable. This is regenerative because there's positive feedback here, and then it is this membrane excitability that drives the cells, that makes the cells hyperactive. And then once, and unsociable, and once they hit a blood vessel during this twitchy hyperactivity, they can spread anywhere they want. So it's a bit like epilepsy. You know, our vision of this complex metastatic process is like epilepsy. Cells become hyperactive in order to behave aggressively. Okay, so now, the voltage-gated sodium channel has two properties that make it clinically amenable, exploitable. And these are not things I'm going to deal with today. I just want to put it in for completeness. One is the fact that the channel is, in fact, a neonatal splice variant. Now, you will know that cancer tissue is dedifferentiated. Many genes therein are expressed in their embryonic forms because of the loss of the normal characteristics of the tissue, and so does we didn't discover that, that's well known in the field, and so does this voltage-gated sodium channel. It's an embryonic form. And it is, we've mapped out its molecular structure, we know exactly what exon is spliced and so on, and it is one of our targets clinically. The other one, which really excites us, and this took some detective work to sort out, in hindsight turns out to be simple, as all, in my view, good science is at the end of the day. And that is that under hypoxic conditions, and I did highlight that growing tumors do become hypoxic on the inside, these channels do not inactivate. Then this persistent current develops, and this is what brings in the sodium, which then does the rest of the kind of pathophysiology. And this is one of our, and I'll show you some evidence here, that because by blocking this persistent current with existing drugs, we can control metastasis, and we're now getting ready for, hopefully, clinical trials. So, in vivo evidence, beginning with mRNA studies, mRNA-level studies. So we've done this, we and others, it's a royal we, initially on prostate cancer. So we have the dominant channel there is NaV 1.7. If we compare in human tissues, prostate cancer and non-cancer, by which non-cancer I mean things like benign prostatic hyperplasia, BPH, because that's what you get in the clinic. And immediately we see that the channel level is much higher, significantly higher in cancer versus non-cancer. We can dissect this further and look at low-grade tumors versus high-grade using pathological criteria like Gleason grade. And again, we see that the expression of the channel goes in step with the expression of the channel, NaV 1.7 mRNA. Now, the way to formalize those data is in the form of a so-called receiver-operator characteristics, ROC, which is a plot of the sensitivity against the specificity of your biomarker. And if the data points fall above the 45-degree line of equality, it means your biomarker is clinically viable as having sufficient sensitivity and specificity to serve as a biomarker. So this is the plot from prostate, and this is a similar comparable plot from the laboratory of Will Brackenbury up at York, formerly at Imperial. Same data for breast cancer. So it looks like, I'm not going to talk about the diagnostics today, but the sodium channel is a diagnostic tool in its own right. Now, here is something we did. Now, it's well known that breast cancer, the first thing it spreads are the lymph nodes underarm. So, pathologists will do a sort of ultrasound to see if there's any spread there. So we did one of those, nerve-wracking experiments. We got in the first run, and we've done it several times now, we got from our pathologists tissue from the breast tumor, okay? And then they also told us if there was lymph node metastasis or not. And so from the tissue, we extracted the RNA and looked for the sodium channel, which in breast is NaV 1.5, and then looked to see if they correlate. Obviously, they correlate, otherwise I wouldn't be presenting the data. The important thing is, this was done blind. I can tell you, it's one of those nerve-wracking experiments, because I held the two envelopes, one of them for the 20 cases, giving, whether the channel was present or not, and the other one whether there was lymph node involvement or not. And these are the data. This is published. In 75% of the cases, there was direct correlation. If the channel was present, there was lymph node metastasis. If the channel was absent, there was no lymph node metastasis. In the 25, an interesting subpopulation of 25%, the channel was present, but no metastasis. And we managed to follow some of these to see that they developed metastatic disease later. So this is good news. That means the channel is actually expressed very early in the metastatic progression. We've never seen a case of metastasis when the channel was not present. In fact, other people in this field have stated that the expression of the sodium channel is necessary and sufficient for invasion to occur. So that is all you need. This fits in very nicely with a paper from Georgetown University, from the group of Norman Lee, driven by Carrie House here. They discovered the sodium channel, again, like breast, NaV 1.5, and they demonstrated, this is published, as you can see, that the channel is indeed in the network of genes, of all the genes controlling colon cancer invasiveness, and I can absolutely assure you, colon cancer metastasis is a horrible disease. It spreads to liver, and it really is horrible. So they knew the genes to look at, and they include things like calcium, MAP kinase, the blockbusters of cancer, mechanism, and so on. And look where the sodium channel turned up. It is actually the first domino in the invasive network, just like the previous breast data suggested. So the channel is expressed, and it's sitting there, and this metastatic army starts marching. And that is what we saw in the breast biopsies, and this is what ultimately was also said for colon cancer. So it looks good at mRNA level. And again, more data, in part from Will Brackenbury's group, showing that NaV 1.5 expression, when people died from breast cancer, where the expression was higher in patients that died, didn't survive versus those who did, who are still alive. And also in recurrence, people with high levels, women with high levels of expression survived, the disease recurred more than those that didn't. And this is the overall survival, and you can see in these analyses, in breast and colon next door, and the channel expression has been divided into high and low. I like to be quantitative. These guys just sort of, fudged it a bit as high and low. And the take-home message is that when the channel expression is at a high level, patients survive less time, breast and colon. That's it. So at mRNA level, it looks good. You can take the fresh tissue into a pathology lab and determine in a prognostic setting what the cancer will do. Now, this is very important for something like prostate cancer, where it is very difficult to use things like PSA and so on to know what the cancer will do. According to our hypothesis and our results, if the sodium channel is present in some prostate pathology, that tumor will ultimately metastasize. That will justify the necessary treatment, including what may become radical surgery. Because often men have paid a high price for radical prostate surgeries by having the prostate removed and then, starting to suffer from incontinence and losing the quality of their life in terms of their sexual activity and so on. Protein level. Now, here is a problem because there aren't very good antibodies for voltage-gated sodium channels. One good news is this neonatal channel, the embryonic channel, because of its really nice special molecular structure, we have made a polyclonal antibody for it, and we can use it on tissues, and we're now in the process of making a monoclonal for it. Here are some immunohistochemistry pictures. At the top, that's prostate. So that's normal prostate. You see this nice regular duct out of which the ejaculate comes out on a good day. And here is the prostate cancer, high grade. You see the duct completely collapsed. And look at the amount of sodium channel, the brown stain there. It goes hand in hand again with the pathology. Breast, again, this is normal breast, nothing, lot of sodium channel in breast. And just to convince you that in the normal breast, when there is no staining, it's not because there is no tissue, there is plenty of tissue there as well. So it does look good. Now, and here is colon. Look at the amount of sodium channel in there. It's filthy full of this embryonic channel. And here is normal mucosa with nice regular epithelial structure and so on. Now, we have done an initial study, this is unpublished, doing the survival analysis, this time at the protein level, and we're beginning to get nice results that patients expressing sodium channel survive much less than those without the sodium channel. So it fits in together. Now the question becomes, not the question becomes, the follow-on question is, what do these proteins do? Of course, sodium channels, like other channels, generate electrical signals. So, and I'm very happy to show here another slide from Will Brackenbury's lab. These are recordings in vivo from tumor slices, breast tumor slices, and you can see these tumors in vivo are generating some kind of action potential spike activity. They haven't done enough pharmacology to know if these are sodium spikes or whatever they are, but these tumors are electrically active. And we're waiting for Sofian to actually come with a kit whereby we can detect these signals in the human body. I'm not kidding. We're very excited by this research, Right, so at protein level, we see the protein expressed by immunohistochemistry and also beginning to see its functional activity as well. Now, tissue electrolyte studies. What happens when sodium channels are active? Sodium comes in. Now, you will know from your basic Hodgkin-Huxley electrophysiology that the transient current normally allows picomoles of sodium. But the persistent current, which develops under hypoxic conditions, will let in a lot of sodium. And we know mechanistically what the consequences of that are. It's all to do with acidification of the tumor surroundings. So can we see this sodium clinically in the tissue? The answer is yes. And here is the two examples here. At the top, you see these are not our data. This is MRI. So we're looking at women with breast cancer, imaged for sodium, tritiated sodium with MRI, and you see semi-quantitatively how sodium builds up in these advanced cancers or metastatic tumors. This is from prostate. The technique here is a slight variation of MRI. This is spectroscopic MRI, MRI-S. And again, the normal tissue, the tumor, this is prostate cancer, the sodium level in cancer is significantly higher than the region next door, the normal. And all these are kind of statistically significant. So it looks like the consequences of channel activity, it does actually reflect itself in terms of these electrolyte changes. And these things can be quantified. These are the breast data showing, again, benign, the level. But here, look, there is quantitative data in terms of millimoles and things, benign, as well as some unrelated tissue, low levels of sodium, and that's it. That's malignant breast cancer. And this is itself a diagnostic marker. It will tell you what the channels are doing and how they might respond to treatment as well. So we are pursuing sodium MRI as a potential clinical method, imaging method for monitoring disease progression and response to therapy as well. Animal models. Anyone wants to see some data from animals? Now, there are several studies now. We did the initial study on a rat model. This is prostate cancer. These are, and this is lovely for Sofian, because these rats are known as Copenhagen rats. So it's just a breeder rat which originally spontaneously developed a prostatic tumor, and by injecting them, and these have been serially passaged, and now we have syngeneic cell lines that are strongly metastatic and non-metastatic and so on. So in this model of metastasis, cells are injected subcutaneously, and they form this primary tumor, some details here, and these tumors then spread to lung. And we see in these discrete blobs the metastases, and we can count them. And I should say, these counts are again done blind. So the person counting the metastases in the lungs do not know whether the lung came from a control animal or a treated animal. Now, we designed this experiment because the sodium channel blocker, what's the best voltage-gated sodium channel blocker in the world? Petroletoxin, God's gift. So, but it is lethal, of course. So we couldn't give it systemically. So TTX was injected into the tumors to make sure of, one, that it reached the tumor. Second, that its action was specific, no side effect, no funny business. And third, by making it local, it didn't create systemic lethality. When we did that and counted the metastases, this is what we got. The number of metastases is halved. This is an early experiment because in those days we could just about through the ethics look at survival. These days you're not allowed to keep the animals beyond a certain number of days. And we could see indeed that by reducing the tumor burden, metastatic burden, the animals survived about 25% longer. So it works. More recently, and this has just been published, we used the same model. This time, instead of TTX, we systemically gave the animals ranolazine. Ranolazine is a blocker of the persistent current. It's an anti-angina drug, which we have repurposed as a potential anti-metastatic drug. And the data are sort of non-parametric, so I'm plotting them here as presenting as box plots, and we can see at both concentrations there is significant reduction of the number of metastases. I emphasize, I trust these data because of the blind manner in which they were obtained. Here are some data from our other friends in Tours in France. They did two things here. So this is another model of, this is breast cancer metastasis. It's a so-called tail vein model, so the cells are injected into the tail vein. It's a bit of a cheat, actually, because you're sort of bypassing half the metastatic cascade by going directly into the circulation. Will Brackenbury did a much more robust version of this, but I want to put it in because there's some pharmacology here. Now, at the top, you see the control data. So you see how these animals, given a couple of months, 2 to 3 months, develop metastases. This is all imaged. And look what happens once the sodium channel is silenced with short hairpin RNA. Complete suppression of metastasis. You know, the animals are completely cleaned. And at the bottom you see the same, in a similar experiment, this time animals given systemically ranolazine, this anti-angina drug. And there we are, we can look at the average data. The short hairpin completely collapses the metastases, ranolazine, not quite, but still highly significant reduction. We've done quite a bit of work with ranolazine now. So this shows its effect on the transient current. Because what we want to do is we want to block this persistent current, this cancer-promoting persistent current, but leave the transient unaffected. So your heart can beat and your nerves can conduct. And that is how ranolazine has been designed. And indeed, also in these breast cancer cells, the kind of concentrations, actually this is an overestimate, the kind of concentration is less than 10 micromolar here. There is insignificant effect on the peak current. So that's why it is a successful anti-angina drug, and we're hoping that it will be equally successful as an anti-metastatic drug. Okay, now clinical studies. Here things get a bit more murky. Basically, what people have asked here, this is not our work, is, look, there are people in this world who take sodium channel blockers, blocking drugs in the form of anti-epileptics, anticonvulsants, anti-angina, all these things. What happens to their cancer? Now, I could find 3 studies dealing with this. Now, these are not, okay, I mean, they are anti-epileptics and anti-da-da-da, but they're not clean like TTX in the way we designed that Copenhagen rat experiment, very clean experiment. Nevertheless, 3 papers, 3 so-called meta-analyses, one from the group of Brackenbury, who actually found an inverse correlation. The patients who were taking these drugs actually fared worse in their cancer. There was a lot of discussion about what was the cause of cancer, where they're dying from the cancer or where they're dying from the whatever disease the medication was used for. The other two studies came up positively and indeed showed an inverse association between use of sodium channel blocking drugs and cancer occurrence. And I just want to highlight one of these. This is from MD Anderson. So you would tend to take the data from one of the world's most well-known cancer centers more seriously. And this is showing survival in terms of months amongst breast cancer patients. These are actually receiving radiotherapy as well. So this is a nice variational theme. The sodium channel usage, or at least anti-epileptic drug usage, is being combined with radiotherapy. And that's something else to bear in mind as we do combination therapies. The take-home message is, indeed, that those who take anti-epileptic drugs survive significantly longer than those who do not in this sort of condition. So on balance, and we're just finishing a mega review on this, on balance we think, yeah, the clinical evidence there is more to do, but the clinical evidence is at the human level is quite positive for us. Now this is something that we wrote because people have noticed, surgeons have noticed, that depending on the type of surgery you use in a cancer operation, the chance of recurrence changes. So in this study, and you can see the data here, women were treated with a local anesthetic as opposed to a general anesthetic. Of course, local anesthetics are also sodium channel blocking drugs. And look, if you treat the patient, the tumor, with a local anesthetic, the chance of recurrence is much less. Local anesthetic versus general anesthetic. So there's an ongoing clinical trial now actually testing, proving this properly. And this may define through the ion channel work how anesthesia ought to be administered for cancer patients with a view to preventing or minimizing the possibility of recurrence. Okay, so this is my sort of summary. The sodium channel offers several advantages as a clinical clinical viability. First of all, we have seen that its expression is upregulated at mRNA level more than 1,000-fold. This upregulation is maintained at protein level and at signaling levels, and it's functional because it promotes invasiveness and so on and so forth, a whole variety of other things. Importantly, the channel is neonatal, so it is targetable. It may even be cancer-specific. and it's what I call systems biology is compatible with expression early in metastasis. Something I haven't mentioned, but we published 2 papers on this, both on breast and prostate. It is under positive feedback control. So the channel is actually self-perpetuating. When it was designed, this channel, whoever designed it, basically said, look, I'll put a channel in there to make cancer aggressive, and I'll make sure the more this channel is active, the more it is expressed. You know, one way of making cancer aggressive. Not realizing that all you would need to do is to block the channel and also block its expression. It's a good position to be in, activity-dependent regulation. And now this is only at in vitro level, and then time will tell about the in vivo and standby patients being treated for time. Okay, so finally, therapeutic potential. There is the possible application of existing voltage-gated sodium channel blocking drugs. We favor these persistent current blockers, and we have the sodium channel antibody. Taken together, where we've got to is we need to design properly and properly design and conduct clinical trials with even the existing drugs. And that is what we are planning as we speak. So that's it. That's what we've done. It was nice to come and do an update and see many old friends and what I call my extended family, Sophia. Thank you.
Thank you, Mustafa, for a fascinating update. Any questions? Yes.
So many thanks for the exciting presentation of the story. For the recurrence issue of the local anesthetic use and the reduce, I wonder whether, unless the drug stays in the body for a while, how it can be related to the feature, unless the drug leaves a memory or the initial blockade by the local sodium is good enough to have a sustained anti-cancer benefit. I just wonder.
Okay, good question. The situation seems to be this. The surgeons call this showering. So what happens is during cancer surgery, when you're removing a lump of tumor or whole breast or something, some cells shower. They actually spread. And it is these that lead to the recurrence. Of course, giving the local anesthetic keeps prevents or reduces that showering effect. So and then the rest, depends on time, what happens.
If you do a search on TGCA, then actually high NAV 1.5 expression is positively correlated with survival. How do you explain that discrepancy in these data sets with your data?
Hold on a minute. So if you do a survey on TRIP-C, you said? No.
On NAV 1.5, expression is positively correlated with survival. From your data, you would expect the opposite.
Well, I showed 3 such studies, and one was positive, suggested positive, but there was a lot of, caveats about that. And the other 2 studies showed a negative correlation.
So how do you explain that? If NAV 1.5 is really such a good biomarker that predicts the metastasis and survival of patients?
I'm not sure I'm following, but all we're saying is if the channel is there, the cancers will progress.
Well, that's not borne out by the genetic data, by the human data.
And which data are you referring to?
Yeah, just look at TCGA. That's kind of the reference database where there's about 1,000 patients with invasive breast cancer.
Oh, I haven't done that. I would love to look at that. Sorry. TCGA. Okay, yeah. It's there, is it?
Yeah.
Are you interested in this field? I'm delighted. I mean, you seem. No, seriously.
One more question. We've just got a few minutes.
Okay.
Beautiful talk, thank you. So as far as I know, one distinct feature of 1.5 is that its activation is reversibly, so its mechanical force reversibly activates 1.5. That's in contrast to other NAVs. Do you think this may have anything to do with the association with tumor invasiveness?
Are you referring to the fact that NAV 1.5 has some mechanosensitivity? Is that what
It does, yeah.
Yes. Now
Under conditions of stress, its
Yes.
activation is shifted to...
Indeed, we are aware of that. We haven't done anything with it, but we've been talking with David earlier. There's no doubt that metastasis involves a lot of mechanical effects, especially during intravasation and extravasation, when the cells go into circulation and come out. And somebody has actually worked out that they are, intra and extravasation are the most critical limiting factors in metastasis. In other words, you know, if it can get into the circulation and survive and come out, those cells will metastasize most. Yes, you know, this is a relatively young field. And it took us enough time to convince our conservative oncologists to take this channel seriously. So I take all the points. There's a lot to do. And of course, it's great news for young people to get involved in cancer and use their ion channel knowledge in a different way, in a situation that is most desperate for new ideas, new drugs, new clinical management methods.
Great. Good. Let's continue the discussion at lunchtime.
Okay.
Thank you very much
Thanks,
again,
David.
Mustafa. Thank you.